Up until the 1970s, the DNA that lies within each of our cells has descended from the original Adamic DNA that God himself fashioned. That DNA, that genetic book, has been a read-only disk. You might say, just as some computer disks are read-only. All that changed when Har Gobind Khorana and his fellow researchers assembled a tiny 207 base pair gene fragment. Although the task is repeated now by students in schools all over the country, it was a monumental, huge step at the time, and our God-written DNA book had now switched from read-only to read-write. Less than 20 years after Khorana's giant leap, the first completely synthetic virus type A was built. Remember this when you hear of new strains of viruses that suddenly appear. Ask yourself if this new strain might be naturally occurring or if it's a deadly clone intended for a target host group. By 2002, an infectious synthetic poliovirus emerged, and this is an important breakthrough because the builder, Eckhard Wimmer, found the poliovirus sequence published online, and he ordered the raw materials through the mail. In 2006, an extinct virus that had died out, according to science, some 5 million years ago, was reconstructed using these synthesizing techniques. So if you have a laptop and a bank account, you can access genomic sequence information and order the nucleic acids and synthesizer for a do-it-yourself Frankenstein kit. This time, you're not using arms and legs from corpses, but the very essence of humanity, the language of our instruction book. You might even call it the key to life. And you might cut and paste your own little Frankenstein monster, complete with as many mommies and daddies as you like. The group of 4 authors who wrote the Venter Institute report mentioned earlier, foreseeing the risk for intentional or unintentional release of a synthetically derived organism. Whether such a release comes from a terrorist cell, a college lab, or a biosynthetic equivalent of a crack house makes no difference. The result would be the same, and any such synthetic organisms, particularly highly infective synthetic retroviruses with the capability of inserting artificial genomic material into host cells, must be considered extremely dangerous. This is a growing industry. The current market globally for just the reagents used in synthesis has grown to $1 billion, and that figure is climbing every day. This industry doubles its activity every 14 months. That's an interesting number. And that number is several years old. The Obama administration has issued a challenge to scientists to reduce the cost of DNA analysis and with that lower the cost of production as well, with the promise of cheaper medical diagnosis and treatment. However, as the process becomes affordable to anyone with a laptop, it also becomes much more volatile. Designer pathogens would be the most difficult to fight, since its sequence would be entirely novel. However, since labs now have machinery that can read the code quickly, sequencing any novel pathogen should be simpler than at any other time in history. This does not, however, ensure our capability to fight this novel attacker. And if a new virus that does more than infect emerges, if it rewrites the human code, then what? How then can any government protect its citizens from such a scenario? What should the government's pre, peri, and post response be? Well, here's what I foresee. If an infectious agent, such as a synthetic chimera, were to emerge, then the government would need a means of determining infection. In other words, they would require a baseline DNA reading on every potential victim. Is it possible then that our government has plans to create a comprehensive database of our DNA? In May of this year, the House voted by a huge majority to approve the Katie-Sepik Enhanced DNA Collection Act of 2010, which provided federal funds to states to collect DNA through blood samples from everyone arrested. Not everyone convicted, but all who are arrested. This was passed in a rare suspension calendar vote, more often used to unanimously approve non-controversial bills, and this procedure precluded all debate. The result is that the federal government expanded their FBI database of our DNA. And in 2008, President George W. Bush signed S. 1858, known as the Newborn Screening Saves Lives Act of 2007. The bill required all newborn babies to have their DNA taken, read, and entered into a database. This DNA can then be cloned and used in countless experiments. This will also serve as baseline DNA, they tell us, for future screenings. And if you think you have privacy, consider this. Your DNA can be lifted from a cup you've tossed into the trash, from a cigarette butt, from skin cells on a tissue, or donated clothing, a hair, or even used prophylactics. Also consider this news item from May 13th of this year. According to the Genomics Law Report, DNA spit kits were to go on sale at Walgreens beginning this year. But the FDA stepped in to stop the partnership with the genetics testing house, Pathway. Pathway sells its saliva collection kits to doctors, who then send the kit home with a patient who collects the sample and mails it into Pathway for a genetics profile. I'm betting that Pathway would love to circumvent the doctor-to-patient pathway and deal directly with the public, which is why the company had tried to partner with Walgreens in the first place. But since the FDA is the roadblock, perhaps the federal agency might make a deal to permit the partnership, provided that Pathway sends a copy of all the genetic reports to the federal government. And if someone wants to get a report on your DNA without your knowledge, couldn't he or she buy such a kit and use your discarded saliva from that paper cup, or perhaps a simple kiss? You know, if a woman wanted to know if male A would be a better genetic sperm donor than male B, then such spit kits would make it deceptively simple. These are just a few examples of ways that your DNA and mine can be collected and cataloged. In a few years, perhaps even today, the federal government will have a complete database of all of our DNA. Let's then assume that a governmental response to a synthetically derived novel virus is to test all victims for DNA abnormalities. Are we infectious? Must we be quarantined? Have you seen the movie Quarantine? In it, a novel rabies virus runs through the inhabitants of an apartment building in New York, turning the humans into something new. The virus alters their native DNA, rewriting the code and changing them into ravenous beasts. You know, the book of Revelation speaks of beasts in chapter 6, verse 8, where the rider on the pale horse called Death, and Hades is given power to kill with sword, hunger, death, and with the beasts of the earth. Now, let's get back to something I told you earlier. Hades has been mentioned before by me. Do you recall when I spoke about the Cumaean Sibyl? Well, remember I also said that she was supposed to stand at the gate next to Hades. Mary Shelley and her apocalyptic vision, ostensibly based on prophetic writings, discovered in a Sibyl's cave, a cave reputed to be an entrance to hell, and how this courtesan to Apollo predicted an ending due to a plague or a tiny beast, a chimeric virus that has the potential to rewrite human DNA, an age-old project that reaches all the way back to Eden when the serpent tempted Eve and she ate, forever changing her and making her fallen. Did our DNA crumble then? Or did it begin in Genesis 6 when the sons of God used their own ancient knowledge that might have been rediscovered in our 20th and 21st centuries, to reshape the very code book of our humanity?